Guys, welcome. This is the second part of the protein biosynthesis. And if you haven't watched the first part, please go and look at it before coming here, because some concepts which we discussed in that video will become very instrumental in this video. But in this video, we'll be looking at the steps, the actual steps involved in protein biosynthesis or translation, that is initiation, elongation, termination. Then we'll be looking at the functions of various factors which are required in the initiation, elongation, and termination. And we'll be looking at some protein biosynthesis inhibitors. Okay, so welcome all. My name is Wallace Munyigi, and I'm the Doctor. Now, let's start with the steps in translation. Now, the process of protein synthesis translates the three-letter alphabet of the nucleotide, that is the codons of the messenger RNA, into a 20-letter alphabet of amino acids that constitute the proteins, the polypeptide chains. Now, the messenger RNA is translated from its 5' end towards its 3' end, producing a protein synthesized from its end, the amino terminal end, towards the carboxyl terminal end. Now, prokaryotic messenger RNA have several coding regions, so they can code for, you can find one messenger RNA has different coding regions. So it is said to be polycistronic, okay? Can carry more than one gene. Each coding region has its own initiation and termination codon and produces a separate species of polypeptide, okay? In contrast, each eukaryotic messenger RNA has only one coding region, that is said to be monocistronic, but we have very few eukaryotic messenger RNA which are polycistronic. But polycistronic messenger RNAs are present in prokaryotes. So as not to forget that PP, prokaryotic messenger RNA, polycistronic. Now, the process of translation is divided into 3 steps: initiation, elongation, and termination. Eukaryotic translation resembles that of prokaryotes in most aspects, but you have some differences. And one important difference is that translation and transcription are temporarily linked in prokaryotes, with the translation starting before transcription is completed as a consequence of the lack of the nuclear membrane in prokaryotes. Remember, in prokaryotes, they don't have the membrane. So the ribosomes can access the messenger RNA as it is being transcribed. But in eukaryotes, you have a nuclear membrane. So transcription will occur in the nucleus, but translation will occur in the cytosol. So the messenger RNA has to be transcribed first, then it will move from the nucleus into the cytosol for translation to occur. Now, let's look at initiation. Now, okay, why is my pointer not moving? Now, initiation. The initiation of protein synthesis involves the assembly of the components. We have to assemble the components of the translation system before peptide bond formation occur. Now, these components will include the 2 ribosomal units, the large and the small subunits. Then the messenger RNA to be translated. Then you need the amino acid transfer RNA specified by the first codon of the message, that is AUG. Then you also require GTP, initiation factors that facilitate the assembly of this initiation complex. And noting that in prokaryotes, we have 3 initiation factors: initiation factor 1, 2, and 3, whereas in eukaryotes, there are usually many, and they are designated as a eukaryotic initiation factor to indicate their eukaryotic origin. Now, eukaryotes also require ATP for initiation, but prokaryotes don't require ATP. The following are 2 mechanisms by which ribosomes recognize the nucleotide sequence, that is the AUG, the start codon that initiates translation. So the first way in which the ribosomes recognize the initiation codon is by use of the Shine-Dalgano sequence. Now, in E. coli, we have a purine-rich sequence of nucleotide bases, which is known as the Shine-Dalgano, the SD sequence, which is located 6 to 10 bases upstream of the initiating AUG codon on the messenger RNA molecule. that is near its 5' end. Then the 16S ribosomal RNA component of the small ribosomal subunit has a nucleotide sequence near its 3' end that is complementary to all or part of the Shine-Dargano sequence. Okay, I will show you that in this diagram. Now, therefore, the 5' end of the messenger RNA will bind to the will complement the 3' end of the 16S ribosomal RNA on the 30S ribosomal subunit. Okay, when I say 30S, you already know that is we are dealing with prokaryotes only, because in eukaryotes it is 40S. Now, and can form complementary base pairs, facilitating the positioning of the small subunit, the 30S subunit on the messenger RNA in close proximity with the to the initiating AUG codon. Now, this is the 30S ribosomal subunit. Then, remember we said within the 30S ribosomal subunit, we have its components, we have the 16S ribosomal. Then, you can see near the 5' end of the messenger RNA to be transcribed in this one, we have the Shine-Dargano sequence. Now, this Shine-Dargano sequence is complementary to a sequence found in the 16S ribosomal RNA near its 3' end. Okay, and by doing so, it will recognize, by recognizing the Shine-Dargano sequence, it will know that AUG is within the vicinity. Then, that was in prokaryotes. That is how prokaryotes will recognize the initiation sequence, or the initiation, let me say the initiating codon. Then, how do eukaryotes do it? Now, eukaryotic messenger RNA does not have the Shine-Dargano sequence. Now, in eukaryotes, the small 40S ribosomal subunit, aided by members of the eukaryotic initiation factor 4 family of proteins, usually bind closely to the cap structure at the 5' end of the messenger RNA, the 7-methyl guanosine cap, and then it will move in the 5', 3' direction along the messenger RNA until it will encounters the initiator AUG codon. Okay, so there will be a region which is untranslated at the 5' end. It is known as the 5' UTR, 5' untranslated region. Okay, and that is the region where the 40S subunit will move along without translating. Now, this scanning process requires ATP. Then you also have cap. Cap independent initiation can occur if the small subunits bind to internal ribosome. It binds to an internal ribosome entry site close to the start codon. Okay, so you have two mechanisms. It is either cap dependent or cap independent. Now, note interactions between the cap binding initiation eukaryotic initiation factor 4 proteins and the poly A tail binding proteins on the eukaryotic messenger RNA usually mediate the circularization of the messenger RNA, and this will prevent the use of incompletely processed messenger RNA or in translation. So remember, that is your messenger RNA. This is the 5' end, and this is the 3' end. At the 5' end, you have a cap, the 7-methyl guanosine cap. Then at the 3' end, you have a poly A tail. So what the initiation factor for eukaryotic initiation factor 4 they usually do is that they bind to here at the cap. Then they will also associate with the poly A tail, and this will circularize this messenger RNA to become a circle. And why must they associate with the poly A tail? So as to prevent the use of an incompletely processed messenger RNA. Okay, so messenger RNA which haven't undergone a post-transcriptional modification cannot be translated. Okay, this in eukaryotes, but in prokaryotes, remember, we don't have post-transcriptional modification. Now, initiating codon. If you have looked at the other video, you must have an idea. Now, the initiating AUG is recognized by a special initiator, transfer RNA initiator. So, a special initiator transfer RNA. Recognition is facilitated by initiation factor 2, which is linked to GTP in the eukaryotes, and eukaryotic initiation factor 2 so linked to GTP plus additional eukaryotic initiation factors in eukaryotes. Now, the charged transfer RNA, the initiator transfer RNA, which is linked to methionine in eukaryotes and methionine in prokaryotes, is the only transfer RNA to be recognized by the eukaryotic initiation factor 2. And it is the only transfer RNA that goes directly to the P site of the small, I think here should be to the large subunit, the large ribosomal subunit, the P site on the large ribosomal subunit. Note that we have some base modifications which usually distinguish the special initiator transfer RNA from the transfer RNA used for internal AUG codon. So even if you have internal AUG codons, which we code for methionine, the first one, the transfer RNA which will initiate, it has some base modification which will enable it to be distinguished from the others. Now, in bacteria and the mitochondria, the special initiator transfer RNA carries an N-formulated methionine, F-met, and after methionine is, how is F-met synthesized? After methionine is attached to the special initiator transfer RNA, a formyl group is added by the enzyme transformylase or the formyl transferase, which uses N10 formyl tetrahydrofluoride as the carbon donor. Okay, its reaction mechanism is the same as the other one which we saw in building biosynthesis, the de novo synthesis of purines. Remember here we had two reactions being catalyzed by the transformylase. Then in eukaryotes, the special initiator transfer RNA carries a methionine which is not formulated. Now, in both prokaryotes and eukaryotic cells, this N-terminal methionine is usually removed before translation is completed. Okay, then the large ribosomal subunit then joins the complex and the functional ribosome, and the functional ribosome is formed with the charged transfer RNA in the P site. Okay, then the A site is empty in this case, and that is where a new amino acid tRNA will come to join. Then specific eukaryotic initiation factors function as anti-association factors and prevent the premature addition of the large subunit. Okay, the GTP on the initiation factor 2 actually gets hydrolyzed to GTP, and in eukaryotes, the guanine and nucleotide exchanger factor is by the eukaryotic initiation factor 2B and facilitates the reactivation of initiation factor, eukaryotic initiation factor 2, which is linked to GDP through replacement by replacing the GTP by a GTP. This might sound very mouthy, but I will show you that in a diagram. So this is what happens in prokaryotes. You see, once methionine is added there, we formulate it. Okay, we add a formyl group, and the formyl group donor is is the N10 formyl tetrahydrofluoride. Reaction catalyzed by a transformylase. This is the F-met formyl and formyl methionine. Okay, and it is the special initiator transfer RNA in prokaryotes. In eukaryotes, it's methionine. Now, how does that was initiation? Elongation. Now, the elongation of the polypeptide involves the addition of amino acids to the carboxyl end of the growing chain. Now, delivery of the amino acid transfer RNA whose codons appear on the next messenger RNA template in the ribosomal A site, also known as a process known as decoding, is facilitated by in prokaryotes is facilitated by the initiation factor TU, which is linked to GTP, and the initiation factor TS, and it requires GTP hydrolysis. Note in eukaryotes, comparable elongation factors are initiation factor 1 alpha, which is linked to GTP, and elongation factor 1 alpha, which is linked to GTP, and elongation factor 1B, 1 beta gamma. So this initiation factor 1 alpha GTP is the same as this one in prokaryotes. This one is the same as this one. Now, both elongation factor TS and elongation factor 1 beta gamma usually function in the guanine nucleotide exchange. Okay, so they will remove this. Once this is hydrolyzed to a GDP, it will remove the GDP and replace it with a GTP. Now, peptide bond formation between the alpha carboxyl group of the amino acid in the P site and the alpha amino group of the amino acid in the A site is catalyzed by the peptide transferase, an intrinsic, and which is an intrinsic activity of the RNA of the large subunit. Okay, so in prokaryotes it will be the intrinsic activity of the 23S ribosomal RNA, but in prokaryotes, so that was in prokaryotes, it will be the intrinsic activity of the 23S, but in eukaryotes, it will be the intrinsic activity of the 28S. Now, after the peptide bond has been formed, the peptide on the transfer RNA at the P site is usually transferred to the amino acid of the transfer RNA at the A site. This process is known as transpeptidation. Now, once you have carried out transpeptidation, the ribosome will then advance one codon or 3 nucleotides towards the 3' end of the messenger RNA. This process is known as translocation, and translocation in prokaryotes will require participation of a elongation factor G, and in eukaryotes we use elongation factor 2 and the GTP hydrolysis. Now, translocation causes movement of the uncharged transfer RNA, which was at the P site. Now it will move to the E site, the exit site for release, and the movement of the peptide transfer RNA from the A site to the B site. This process is repeated until termination codon is encountered. However, because of the length of most messenger RNA, more than one ribosome at a time can translate a message. Such a complex of one messenger RNA and a several number of ribosome is known as a polysome or a polyribosome. Now let's look at that. You can see we have a peptide transfer RNA at the P site, then an amino acid transfer RNA will come to the A site, then via the intrinsic peptide transferase activity of the 28S ribosomal RNA in prokaryotes, or the 23S ribosomal RNA in prokaryotes. I'm confusing myself. 28S is in eukaryotes. 23S is in prokaryotes, and those ribosomal RNA are the one which have the peptide transferase activities. They will catalyze a peptide bond formation as shown here. Okay, as shown. You see, we transfer this to the A site, then translocation will occur, and this uncharged transfer RNA will move to the E site, and then this peptide now, the peptide transfer RNA here will move to the P site. Now this is the uncharged one, will move to the E site for release. Now, termination. How will it occur? It will occur when one of the 3 termination codons moves into the A site. UAA, UAG, UGA. These codons are recognized in the E. coli by the release factor. Release factor 1 will recognize both UAA and UAG. And the release factor 2 will recognize UGA and UAA. So UAA is recognized by both the release factors 1 and 2, but UAG will be recognized by the release factor 1 and UGA only by the release factor 2. Now, the binding of this release factor will result in the hydrolysis of the bond linking the peptide to the transfer RNA at the P site. This will cause the newly formed protein to be released from the ribosome. Then, a third release factor known as the release factor 3, GTP, then causes the release of these release factors, okay, as GTP is hydrolyzed. Note that in eukaryotes, we have a single release factor, the eukaryotic release factor, which recognizes all the 3 termination codons. Then a second factor, eukaryotic release factor 3, functions like the prokaryotic release factor 3, and will cause now the release of the eukaryotic release factor, and as GTP is hydrolyzed. Now, the newly synthesized polypeptide may undergo post-translational modification, and the ribosome subunits, messenger RNA, transfer RNA, and the protein factors are then recycled and used to synthesize another polypeptide. You do not degrade them, you recycle them. Note, however, in prokaryotes, ribosome recycling factors mediate separation of the subunits. But in eukaryotes, the eukaryotic release factor and ATP hydrolysis are usually required in the separation of these two subunits of the ribosomes. Now, let's look at the function of these factors. Here we have a step. It is either P for prokaryotic, E for eukaryotic, then the factor itself and its function. Now, which factors are required for initiation? We have the initiation factor 2 and the eukaryotic initiation factor 2. Both are bound to GTP. Both they bring the charged initiating transfer RNA to the P site, and it is the only one which is brought by the initiating factor 2. Okay. Then you also require the initiating factor, initiation factor 3. Both in this E means eukaryotic initiation factor 3, and they prevent the association of the two subunits of the ribosome. Then elongation. In prokaryotes, you have the elongation factor TU-GTP, which is synonymous to the initiation factor, elongation factor 1 alpha GTP, and this one brings the other charged transfer RNA to the A site. So you see only the special initiator transfer RNA is brought to the P site by an initiation factor. The other ones are brought by the elongation factors. Then you also have the elongation factor TS, which in eukaryotes is the elongation factor 1 beta gamma, and it is used in the guanine nucleotide exchange. It is a guanine nucleotide exchange factor. Once we hydrolyze this GTP to a GDP, you need to cleave that GDP and replace it with a GTP once more. That is the function of these elongation factors. Then you have the elongation factor G and the elongation factor 2, which are necessary for translocation, the movement of that small ribosomal subunit at a distance of 1 codon. You require these elongation factors. Then termination. You require the release factor 1 and 2, and the eukaryotic release factor to recognize the stop codons. Remember, I said UAA is recognized by both the prokaryotes. Then UAG is recognized by the release factor 1, and the UGA is recognized by the release factor 2. Then you require the release factor 3 to release the other release factor. Now, protein synthesis inhibitors. All that I've said can be summarized in this diagram, two images. They are the last two images. Now, initiation occurs, initiation factors aid in the formation of the 30S initiation complex. Then the charged initiator transfer RNA is brought to the P site by the P site of the 30S subunit by the initiation factor 2. Okay, and this is what I'm saying. You can see the initiation factor 3 here will prevent the association of the small ribosomal subunit with the large. So what streptomycin does is that it binds to the 30S subunit and it will distort its structure. By distorting its structure, it will interfere with the initiation of protein synthesis. Okay. Then let's go to this other process which is targeted by tetracyclines. They usually interact with the 30S subunit, and by doing so, they will block access of the amino acid transfer RNA to the A site, thereby inhibiting elongation. So tetracyclines will prevent this charged transfer RNA from accessing the A site. Okay. Then we have this other one, peptide transferase activity of the large ribosomal subunit is inhibited by chloramphenicol in prokaryotes. Now, chloramphenicol is in prokaryotes, but high levels of chloramphenicol might inhibit mitochondrial protein biosynthesis. Now, in eukaryotes, the peptide transferase activity in the large ribosomal subunit, which is intrinsic to the 28S ribosomal RNA, is inhibited by a drug known as cycloheximide. Okay, so chloramphenicol for prokaryotes and cycloheximide for eukaryotes have the same working mechanism. Then we have another drug known as pyromycin, which bears a structure. It is similar to an amino acid transfer RNA. As accurately speaking, it is similar to the tyrosinyl transfer RNA, and by doing so, it will be incorporated to the growing peptide chain, and by doing so, it will cause premature termination. Okay, then let's look at the other diagram. We have another drug known as erythromycin, which usually bind irreversibly to a site on the 50S subunit and blocks the tunnel by which the peptide leaves the ribosome, thereby inhibiting translocation. But I had seen some texts suggest that the working mechanism of erythromycin is that it blocks the attachment of the peptide transfer RNA to the P site. Okay, remember tetracycline were blocking the amino acid transfer RNA to bind to the A site. So please remember that. Then we have some drugs which inhibit the protein synthesis in eukaryotes. Diphtheria toxin usually binds to the elongation factor. It usually causes ADP ribosylation of the eukaryotic elongation factor 2. Remember, here it is the eukaryotic elongation. What was the function of the eukaryotic elongation factor 2? Was used for translation. So this one will you will have when diphtheria toxin is present, it will be inactivated. Okay, then you have ricin, which is present in castor beans. It usually remove a specific adenine in the 60S subunit of a ribosome that is in the eukaryotes. It usually depurinate the 28S ribosomal RNA in the 60S subunit of the ribosome, and by doing so, it will prevent protein synthesis. Okay, so I think this was the here we had done. Then we have another one which is known as fusidic acid. Fusidic acid usually prevent the recycling of the transfer RNA. Okay, and it also inhibits another process. Fusidic acid inhibits protein synthesis at 2 points, but one of them was at prevents the recycling of the transfer RNA. So I think that was that. And if you have watched the two videos, I think you have it is of benefit and you have understood. So let's look a summary of this protein biosynthesis inhibitors. We have those which inhibit prokaryotes, those which inhibit eukaryotes, and both. So those which inhibit prokaryotes, they can either bind to the small subunit or the large subunit. So those which bind to the small subunit are either tetracycline or aminoglycosides, the streptomycin. Okay, then those which bind to the large subunit, the 50S, are chloramphenicol and erythromycin. Okay, then those which inhibit the eukaryotes are either diphtheria toxin or ricin. Okay, then pyromycin inhibits both prokaryotic and eukaryotic protein synthesis. Thank you. I hope you have gotten something. So, and please subscribe for more content. Thank you.